Good afternoon, class. Thank you for connecting and following Metric Hub. Today we are meeting actually to start our revision sessions. Term 1, I can say call this one first session of revision, preparing for end of term 1 or consolidating N1 work and ensuring that our progress is so smooth. So without wasting any time, we are going to work on the chapters or pieces of work that are supposed to be done in term 1. As you can see, this is test 1 March. Of course, this is a revision paper that we are working on. You always know that you read instructions when you go to exam. Please take note of that. And without wasting any time, I want you just to go through, just check through what we are going to be working on. I'm sure that you know these pictures in my lectures and classes. I always tell my students that we number them 1 to something according to the progress that we are going through. So please note that these questions, these are technical specific questions that we're gonna meet when we go to final exams, as well as writing end of term. So let's just follow up. I just want you to just have a see-through of what is in this revision paper. We are going to revise all these. Just follow to the end so that we can be able to understand and review and catch up wherever we are behind. So first question, section A, it is the multiple choice question where various options are given. So we choose the best out of the options that are available. The structure in the amniotic egg that encloses the amniotic cavity containing amniotic fluid. I'm sure that you know the amniotic egg, the egg that is able to survive on Earth. We say that this is a shelled egg. I had a lot of presentations on this. We are saying this is a shelled egg, a egg with a shell. That's what we call an amniotic egg. And then the amniotic cavity, that fluid that encloses the developing embryo, which we say that it takes or it contains the amniotic fluid. So that structure, what is it called? I'm sure that you know the yolk is for feeding and ensuring the survival or nourishment of the young one. We talk about the ariontos, we talk about the chorion, but in terms of the amniotic egg, that structure that encloses amniotic cavity, it is called the amnion. It is called the amnion, where in mammals we call it amniotic sac. That is to mean to say the answer is what is C. We have a presentation on the structure of an amniotic egg, and we all as analyzed and reviewed very clearly there. So please, let's check on that. Implantation takes place in the dash. This is in terms of internal fertilization, where we say that the young one, it attaches the fertilized egg, the zygote attaches in the what? It attaches itself in the uterus. It is in the uterus where we see the zygote. It has to implant itself for nourishment, for it to be able to survive, to allow the development of the young one. The placenta, it has to work through the umbilical cord, so it attaches itself, that zygote, to the uterus, inside the uterus, which is the womb. Every other part, we are saying the vagina is a birth canal. It is also for excretion. It is also for copulation, cervix, actually for birth process, which allows or which enables the expansion and contraction during labor pains. The ovary, obviously, we said that it is the one that releases the eggs. So we are saying it attaches, implantation occurs in the uterus, where we talk about the endometrium lining. During copulation, the penis releases sperms directly into the copulation. it occurs, and it is when eggs are released inside the vagina. That's where eggs are released during the action or during copulation. Fallopian tubes, we say that is where implantation occurs. Uterine cavity is for implantation, and then C is the right answer. Give the correct biological term of the following descriptions. We write only the term next to the question number. I always say that please take note of the instructions per question. The type of development within which offspring are born helpless and cannot move or feed themselves. We talk about forms of development in invertebrates. So there is also a presentation that is coming or that has been loaded in on this. So we have got atresia and precocial. Atresia is when the young ones are born helpless. They depend on the parents. They've got they need parental care. They need parental care. They need parental feeding. They need caring from the parents. So it is called atricial development. The opposite of it is precocial. Precocial is those ones which are born able to move and able to feed themselves. They can even run after birth. We say those are called precocial, but the ones which are born helpless are called precocial. And then the part of amniotic egg that provides nutrition to the developing embryo, it is called the yolk sac. It is the yolk sac. The yolk actually it nourishes. It is there for nourishment of the developing young one, which again we have seen that in some uh, context of in some context of evolution, we say that the size of the yolk also determines the level of developing of the embryo, the developing chick inside the egg. So the yolk has to be big enough and it has to be nutritive enough for it to nourish the developing young one. So we say that it is the yolk sac that actually nourishes, that provides nutrition. And then the analysis of DNA samples to identify individuals that may be related, that is also called DNA profiling. DNA profiling. I have a presentation for you on the channel. Please check on DNA profiling questions that arises there. We have got DNA profiles or DNA bars that are given, and you are asked to identify either an adopted child or the criminal in that context. So the likeliness of the profile to look alike to each other defines whether the person is adopted or it is the person is what is a criminal. So we have talked about the types or the uses of DNA profiling quite a lot of times. It is there also on channel. Please follow up and get right of this division of the nucleus. It is called karyokinesis. Karyokinesis is nucleus division. The division of the nucleus is called karyokinesis. Karyokinesis is the division of the nucleus. So karyokinesis, the division of the nucleus. We have to indicate that it applies to A, B, both or none. I'm sure this question paper or this practice paper is actually profiling or showing us the replica of the paper that we write at any other times, like end of the year. You need to know the structure of your paper. We have got types of paper like this one. So we are given this one, which says hatching of eggs occurs in the female reproductive system. Hatching of eggs occurs in the female reproductive system. It is where we talk about ovovivipary. of ovary, we say the eggs hatch inside the reproductive system. External fertilization, the eggs are fertilizing outside the body of the female. So they actually have to have a suitable environment for them to survive, which has to do with extreme vulnerability to predation and so other things. So it is what it is: a site of meiosis in females. If we are in female, meiosis, it occurs in the ovary. Meiosis, it occurs in the ovary, which is B, which is A, of course. So now testis is for what? This is for male. Site of meiosis in what? In male, it is the what? It is the testicles. So in female, we talk about the ovary. And then the next question, the result of an extra chromosome number. The result of extra chromosome number, it is the result of what? The result of extra chromosome number. We say that it is sometimes called trisomy 21. Trisomy 21, it is, we are saying, whereby an individual or a zygote has got 3 sets of chromosome 21. It is as a result of what? Of Down syndrome. Chiasma is the point of crossing over. So A is the answer. Down syndrome is the result of an extra chromosome. It is caused by extra chromosome 21, which is caused basically by non-disjunction of chromosomes during meiosis. Then 2N chromosome is fertilized by an N. 2N zygote is fertilized by an N, which results in 3 sets of chromosome 21. Then that individual, if the egg develops to birth, it results in a young one with a disorder defined as Down syndrome. The diagram represents some processes in terms of protein synthesis. It's some stages or some process of protein synthesis, which again, I'd say that sometimes you would find out that other writers or other examiners, they just give you both uh, transcription and translation combined on a single diagram, where many other questions are therefore developed or are asked. So now what you need to understand is you need to know the difference between the two: protein synthesis and also transcription. Transcription is when the mRNA is formed, which occurs in the nucleus, and then the mRNA exits the nucleus, uh, going into the cytoplasm, where uh, protein synthesis has to take place. So now I've been always saying that you need to know the terminology that applies in this context. So name the overall process occurring in the diagram. It is defined as this is defined as protein synthesis or translation. We are seeing we are seeing tRNA, we are seeing mRNA. The moment the diagram have both of them, and we have got this pairing of codons and anticodons in a diagram, it identifies the production of proteins. That is defined as protein synthesis. Is protein, which we said it's also called translation. Protein synthesis, also called translation, is one and the same thing. The process by which proteins are formed. Give the letter that represents transcription. That's what I've been saying. A letter that represents transcription. Transcription occurs when a piece of DNA it is also pairing with a piece of a small piece of mRNA that is being formed. So we see that C is representing that where the piece of DNA it unwinds, and then we have got a single strand that wedges below it. That wedging of that single strand below the strand of DNA, it identifies the process of what? Of transcription. So transcription is occurring in what? By letter C, is occurring by letter C, as you can see on the diagram there. Single strand wedging below DNA strand, which is the original strand of DNA there, causing the assimilation of the genetic code into the single strand, the mRNA that is being formed, defining transcription. Movement of mRNA out of the nucleus. It moves out of the nucleus through the nuclear pore, which is taking place at E. E is identifying the exiting of mRNA. It is, next time they say, at where does it exit? Name the point or the place where mRNA exits. It exits by the nuclear pore. And where does it go? It goes into the cytoplasm. And then codon-anticodon linkage. That is, we are saying the codon-anticodon linkage, it is therefore which is taking place at A. The codon-anticodon pairing, the codon-anticodon linkage. Here, this side on tRNA, we have got the codons, and then we have got the anticodons on tRNA. We have got the anticodons there in tRNA. Anticodons, we say that they are found on tRNA, and then on mRNA. On mRNA there, we have got the what? We have got the codons. That linkage is what they are defining or they are referring to. So it is taking place at E. It is taking place at E. Sorry, linkage is taking place at A. The linkage of codon-anticodon pairing is taking place at A. Remember to say that that pairing or that linkage is always supposed to be known that is complementary, which I want you to understand that the pairing which is taking place there, it is supposed to be always known that A, it pairs with what? With U, G, it pairs with C. That pairing or that linkage, it is supposed to be always remembered. That's how this relationship or that linking occurs. That's how the linkage takes place. So please, class, take note of that. Don't miss it. It might come like that in the next paper. We are given premature delivery of babies born between weeks 28 to 35 of gestation period. The period of pregnancy has been a concern in many provinces. The care of premature babies is very expensive. That's very true. It's very costly. So now, it is very costly. So what happens is women with a history of premature delivery, women with a history of premature delivery are sometimes given progesterone treatment between weeks 16 to 20. Between weeks 16 to 20. So what the question wants us to understand here is that however, this treatment is believed to lead to development of gestational diabetes mellitus in the mother. The mother will suffer from diabetes mellitus, which is usually happening during pregnancy. The following investigation was done. 300 pregnant women with a history of premature delivery participated in the investigation. Those that had pre-existing diabetes mellitus were excluded. The ones who once had the condition were excluded. The women were divided into two equal groups, A and B. The women in group A were injected once a week with 250 milligrams of progesterone between weeks 16 and 20. These glucose levels were measured and recorded daily between weeks 16 and 36 of pregnancy. Group B was the control. Remember, I said again that you need to understand these experimental questions. It is quite possible for a student to get all the marks here. These are bonus questions again, which are given for us to boost our marks. So then, please don't miss marks there. The question therefore says, why is the injection of progesterone a good treatment to prevent premature delivery? Uh, I want you to remember the essence of hormonal control or the concept of hormonal control in terms of the menstrual cycle and pregnancy. So now, progesterone, it actually maintains or it thickens the endometrium and therefore it helps to maintain pregnancy. It helps to maintain pregnancy. So what happens is progesterone it maintains or thickens the endometrium. It maintains or thicken the endometrium uh, and therefore helps to maintain pregnancy. It helps to maintain it helps to keep the pregnancy. We talked about the thickening of the endometrium. If it keeps taking place, if it keeps going, it means fertilization has occurred. And what is causing that? It is the secretion of what? It is progesterone that causes thickening of the endometrium. So as that happens, pregnancy is what is being sustained. There is the sustenance of pregnancy that comes with the thickening of the endometrium. So these people were injected with it as and it works as a good treatment to prevent premature delivery because it helps to maintain the pregnancy. Identify the independent variable and also the dependent variable. And therefore, if you look at this, we say that you read the question from where the statement says, what is the investigation was done. The following investigation was done. 300 women with history of premature delivery participated in the investigation. However, this treatment is believed to lead to development of gestational diabetes mellitus in the mothers. So we are saying we need to look into this to say the effect of what and what. So the pregnancy, what is being investigated here? What is under investigation? So what we need to understand is that we need to understand where we talk about the issue of progesterone and the development of gestational diabetes mellitus. We are saying an investigation was done in terms of the issuance of or the administration of progesterone treatment and also the development of gestational diabetes mellitus. So now what we are saying is that the independent variable it is progesterone treatment. The independent variable is that there was progesterone treatment. Progesterone treatment is the independent variable. We are saying progesterone treatment was this treatment believed to lead to developmental gestational diabetes mellitus, which is caused because we are saying there is progesterone treatment progesterone treatment is the independent variable. Progesterone treatment it is the independent variable, and then the dependent is development of gestational. The dependent, it is development of gestational diabetes mellitus. Development of gestational diabetes. Development of gestational diabetes is the dependent variable, because we are saying when these women are treated with progesterone, they develop the chances of gestational diabetes. It is as a result of the treatment with progesterone. So treatment with progesterone, it's an independent variable, and then the dependent is what development of gestational diabetes. If people are not treated with progesterone, therefore they cannot have, or they may have less chances of diabetes. So that is how we can work out that. Describe how the investigators determined whether any of the participants in Group A had developed gestational mellitus. So in this case, we are saying, how do they see? Describe how the investigators are determined whether the participants in Group A had developed the condition before. So we see that in Group A, the women in Group A were injected once a week. They were measured. These were glucose levels were measured and recorded. We are saying the glucose levels were taken daily. That is one way. Glucose levels were measured and recorded daily. And also we see that when the glucose levels of the women remains high continuously, indicates that the development of gestational diabetes is possible. So we are saying that is what happened. The women in Group A, we are saying this is what happened there. It is shown that glucose levels were measured. So the answer is that the glucose levels were taken daily. Their glucose levels were taken daily. They were measured daily. And then we said that when the glucose levels of a pregnant woman remains high continuously, it indicates the development of gestational mellitus. When the glucose levels of a pregnant woman remains high continuously, what it means, it indicates the development of gestational, it means development of gestational diabetes. Development of gestational diabetes. That's what happens there in question 2.13, and then 2.14 state one factor one factor 2.14 state one factor that were kept constant when the progesterone was administered in group 1. We are saying state one factor that was kept constant when this was was administered. So when this was administered, what happens is that what was kept constant is that we talk about the same dosage which was given, the same dosage which was given. We talk about the women in group A were injected once a week with 250 milligrams of progesterone. So we are saying one same dosage of progesterone, same dosage of progesterone was it was administered. Same dosage of progesterone. Same dosage of progesterone. And then we can also talk about the same period of time for injection, because you see that from this context, it is given that it was given between weeks 16 to 20. It was given between weeks, we are saying between weeks 16 and 20, which also shows that that was kept constant. So we are saying same period of injection, same period of injection, same period of injection, which we can also need to understand that. And then we can also talk about same frequency of injections, which is weekly, same frequency, same frequency. That is, we are saying that same frequency of injection, which was weekly also. So therefore we said, why were all these done? These were done actually to increase the reliability of the result, to avoid variation. And then question 2.15, it comes and says 2.15, group B was the control. Explain the importance of control in this investigation. The control actually, it is not manipulated, it is not worked with, it is not changed anything. So it helps to, it actually helps to evaluate the correctness of the investigation. So group B did not receive progesterone, was unmanipulated. It was unmanipulated. So what it means is that no progesterone was administered. If gestational diabetes develops in group A, it can be due to progesterone treatment only, such that if gestational diabetes develops in group A, it would be due to progesterone treatment. It will be conclusively attributed to progesterone treatment. It will be due to progesterone treatment. So basically, class, what you need to know is that uh, control in an investigation is always used as a standard of comparison of results so that we can be able to understand the reasons of change or the factors behind the investigation, that they are strictly caused by the factor under investigation. We can be able to evaluate by comparing the manipulated and unmanipulated set of experiment. That is the purpose of the control within an investigation. It is used to make comparative analysis of the results so that we can be able to evaluate the exact cause of the factors that are under the investigation. I guess we are still together. We want to move on to the next question. Next question. It is the structure representing the part of female reproductive system. I said that these are also other bonus marks, which we must make sure that we don't miss as we go to exams. There is no way whereby we can go to exam and people are not aware, or people, they don't know reproductive systems, whether male or female. So let's just go to the question. Provide labels for parts A, B, C, D. Parts A, it is the fallopian tube. We say that is where fertilization occurs. Fallopian tube, which we said again that it is the what? It is the oviduct. It is the oviduct. And then we think label A, B. B is the ovary. And then C is the endometrium. C is the endometrium or uterus lining. Endometrium is C. And then D, we can say D, it is, we can say D is the cervix. That is the cervix there. D is the cervix. We said it is important for birth process. It stretches to allow birth to open up the birth canal, and then give the letter and name of the part in which oogenesis occurs. We said oogenesis is the formation of the formation of female sex cells. So the letter and name, we said that always please note that there are two questions here. These are two questions which are coming from this question here. So you have got 2 marks because you need to give the letter and name. So oogenesis, it occurs in the ovary. So we need to see which part represents the ovary. Like we say that oogenesis takes place. So it is B, which is the ovary, the letter and name. So we say B, which is the ovary, and then to which the embryo is attached during implantation. Implantation, it occurs in the endometrium. So the letter where endometrium takes place, or which identifies the endometrium, it is C. Attachment, it is C, which identifies the endometrium, and then into which the ovum is released for fertilization to take place. It is the oviduct. Oviduct is shown by A. Fertilization takes place in the oviduct A. We say that is the oviduct. We say that it is the fallopian tube. And then why? Next question is saying, why will a blockage at D lead to infertility? Blockage at D a blockage at D. Why can it lead to infertility? Obviously, if D is closed, blockage, it meaning to say that closure of this one, if it is closed, what happens? The closing of the cervix or the passage to the into the reproductive cyst organ, sperms will not reach the ova, and it cannot be in the fallopian tube. The sperm will not reach the ovum to go into the fallopian tube. The sperm cell, when it is released, it will not reach. It has to pass through. Sperm cells has to go through. I'm sure I remember the process of fertilization, how it occurs. Sperm cells, they must compete, they must rush through here. So what we are saying is that if it occurs like that, no sperm cells can pass through. No sperm cells can pass through this if it is blocked. So what it means is that no sperm cells can reach, can pass through to the what? No sperm cells can pass through to the, no sperm cells will pass through to the fallopian tube, to the fallopian tube or oviduct for fertilization to take place. So what we are saying is that for fertilization to occur, so blockage of the passage, it means how would it lead to no infertility? How would it lead to infertility? We are saying it avoids fertilization. It avoids or eliminates chances of fertilization. Obviously, if there is no fertilization, there is no chances of pregnancy. So we are saying infertility, it will be obviously be there as a failure. It will be actually not take place. We are given the diagram represents one cell. It represents one cell in a phase of meiosis. I say it again, please check that. It is also available on channel. It is also available on channel. So the question is saying, I'm saying we should be able to label these to say, I said the first thing when we go to meiosis is being able to label everything. What is all these things on the diagram? There is a presentation also on channel where these were labeled and explained their functions. The first question is saying, identify the phase of meiosis that is shown here. Identify the phase of meiosis that is shown. As we can see, this is metaphase 2. Metaphase 2. I'm saying there is a video or even many of them which are there to address the process of meiosis. Draw a labeled diagram of structure C as it would appear in the final phase of meiotic division. Show the correct shading. So what we need to do here is to show the diagram. I'm saying draw a diagram, a labeled diagram of structure C, a labeled diagram of structure C, a structure C, uh, of structure C as it would appear in the final phase of meiotic division. So we can say structure C in the final phase of meiosis, that is the heading. Structure C in the final phase. In the final phase of meiosis. which is, I'm sure that we know that that is the final phase is what? Telophase 2. Telophase, the final phase is telophase 2. So we can say that this must appear as, we can have to draw as, we must have a chromatide. So that we can say so we have got unreplicated, unreplicated, which we say that is chromosome, we are saying chromatide or daughter chromosome. It's unreplicated. I have got defined this also on channel. It is unreplicated, which is a chromatide. It is a chromatide. It is also a daughter chromosome. Which we say that here we have the centromere. We have the centromere. So the title is that one, structure C in the final phase of meiosis. And then we have got that diagram. It can actually show that that is the diagram, and that is how it looks like. And then the next question is saying, state one difference between metaphase 1 and metaphase 2. But before I go there, I want you to note something here in terms of this diagram. You must be able to give the heading that is proper when it comes to marking of this question. You need to have the heading. It is very important. The heading, which is what I have given here to say, structure C in the final phase of meiosis 2. And then it must have that heading. And then the drawing also must be very perfect, which you must be able to show that in terms of the drawing, it must be clear that you show, in terms of the drawing, you must be able to show as is being represented on board here. So it is very important for you to be able to show that, to say it is unreplicated or it's a daughter chromosome, which we can also say that it's a chromatide, which is separated. And then we also have one shaded, one unshaded, and then we are saying, or one shaded. It can be either of that, one of the either of that structure. You can either draw them as to say one shaded or one unshaded. It depends with your choice of diagram. And then we are saying it should also show those labels that can enable us to ensure that there is, if I was going to draw unshaded, I would just draw it unshaded. And then we say that it is unshaded, which we can say that that can be also correct to say it's another way of doing it, which is also representing the same diagram. So state one difference between metaphase 1 and metaphase 2. In metaphase 1, the chromosomes arrange at the equator in homologous pairs. In metaphase 1, I'm sure that we have seen that in metaphase 1, the chromosomes arrange at the equator. Arrange at equator. They arrange at equator in homologous pairs. In homologous which is also different in metaphase 2, where chromosomes arrange at the equator in singles. Whereas in metaphase 2, chromosomes are arranged as singles. That is one thing that we need to take note of when it comes to these diagrams or this meiosis process. That is one thing that we need to understand. They arrange at the equator single as a chromatide, and then describe the role of spindle fibers in the movement of chromosomes during meiosis. We said that I always tell my class that the spindle fibers are there to affect the stages of meiosis. What they do is that actually the spindle fibers, they shorten to pull the chromosomes or chromatides to opposite poles of the cell. Spindle fibers, they shorten for them to pull the chromatides. The chromatides are pulled to opposite poles of the cell. To opposite poles of the cell. That's the function of the spindle fibers. They shorten for them to pull. We say that as they shorten, they are shrinking, they are becoming smaller, contracting towards the poles. As they contract towards the poles, they are pulling the chromatides to opposite poles of the cell. That's the function of these spindle fibers. And then 3.21, we are given the structure of a DNA structure. We are given the structure of the DNA molecule. And then this one also, it must come as a bonus. I always tell you that please don't miss unnecessary marks. You can't go to exam not understanding DNA structure. Identify part C and D respectively. Part C, if you look there, part C is the sugar, which we say that it is the deoxyribose sugar in that is C is deoxyribose sugar, that is C, and then obviously D is the phosphate. It is the phosphate group. D is the phosphate, and name one type of bond that joins A and B. This we say that the opposite nitrogen bases are held together by what? By weak hydrogen bonds. This red is the weak hydrogen bonds that joins the complementary bases. So it is called weak hydrogen bonds. is the weak hydrogen bonds. And then next question is saying give one visible reason for for identifying the above molecule as DNA. We said that DNA is shown. What identifies DNA, as you can see, it is actually a double-stranded molecule. We have got this strand here, we have strand 1, and we have got strand 2. So together, this one and that one, they come together. As they join together like that, we are saying it's a double-stranded. So what we are saying is that it's DNA because it is a double strand. It is a double-stranded or base pairs. Bases are pairing. Base pairing or bases are pairing. One, we are saying it is a double-stranded. It is double-stranded. It is double-stranded molecule. That means it's DNA. Two, we are also saying that we can say base pairs are present. They are visible. We say they are present in this case. As you can see, we say A and B. This is base A. We say it is A, adenine, and then it is T, thymine, and then we have the weak hydrogen bonds there. It is G, and it is C. So base pairing is visible. It is present, as we can see on this diagram. That is the reason why we say that. And then two uses of DNA profiling. Uses of DNA profiling. One, it is used to solve criminal investigations, to solve criminal investigations. Uses of DNA profiling when crime scenes, when police have got unresolved cases, they can be able to further find the perpetrators through DNA samples. That is very important. We also use them to identify family relationships. Identifying family relationships. Identifying family relationships. DNA samples, DNA profiling can also be used to identify organisms from their tissues. Identify organisms from their tissues. That is very important. It's another very important use of this one. It is also used in research into variation in population. Research into variation. Research into variation in population. That is one other use. And then we can also use it otherwise in terms of testing specific alleles that can cause a genetic problem or matching tissues for transplant. That is another use. Identifying matching tissues for transplant. Identifying matching tissues for transplant. That can be done through DNA profiling. There are quite a lot which we can give in terms of this function. And then tabulate the structural differences between RNA and DNA. Differences between RNA. So we say RNA. The question is saying tabulate the difference between RNA and DNA. So we have to give the differences between RNA. We say RNA is a single strand. DNA is double stranded. We said there are two strands that forms DNA. And then we also say that is RNA, it has got ribose sugar. Contains dioxiribose sugar. And then it has uracil. However, contains thymine. Instead of uracil, DNA has got thymine. So these are the differences. We are saying when they say tabulate, you must be able to draw a difference. I mean, a table that identifies the two points in question, or the two contrasting points of discussion. So please keep following the channel by subscribing, and also click on join and follow the prompts that are there. They really help you in order for you to keep connected. So please keep connected by clicking join and then become a member. It's quite cheap for you. It's quite affordable for all your modules. You are able to follow up all of them through that one single click, no matter how many subjects you are doing. But by joining once, you have got access to all of them. So please do so, because term 2 and term 4, we don't want to see regrets. Thank you very much for connecting, and let's do it again. Have a good day. Thank you.